Let's talk about creating a cladogram based on DNA or chemical evidence of evolution. Hey, after watching this video, you should be able to use a cladogram that's based on a DNA sequence or amino acids to determine relatedness between species and place them on a cladogram. Well, what do you need to already know? What DNA is, that would be good. The bases, how to use a structural cladogram, those are all good starting points. So first, let's talk a little bit about the fact that of the strands of evidence that support the theory of biological evolution, chemical evidence is the one we haven't really talked about yet. And the first big clue is the fact that every living thing on the planet, from bacteria to blue whales, from earthworms to maple trees, uses the exact same chemical, DNA, to transmit their genetic information and directions for how to build proteins. What's interesting is that all known living things also use the same chemical, RNA, in the protein construction process. There's a little bit more chemical evidence than that even, in the fact that DNA sequences between species can be analyzed, and we can look at seeing how many changes there are between two species. For instance, humans and fruit flies, both animals, share 60% of their DNA sequence. That means 60% of your DNA sequence is identical to a fruit fly. Really? 90% of your DNA sequence is identical to a mouse. You're both mammals after all. There are a lot of things that your body does that a mouse's body does too. And by the time we get up to the level of primates, which includes you and the chimp, your DNA sequence is 99% identical. Remember when we did these gel electrophoresis patterns to compare relatedness between two organisms? Yeah, I thought so. We can basically do the same thing with DNA sequences between species to build a cladogram that puts together the evolutionary relationships between species. Let's say we have three species. We'll call them, real creatively, species 1, 2, and 3. And we have a section of DNA that all the species share. But we notice something when we look at it. Species 1 has a C in the 4th position, but species 2 and 3 each have an A there. Well, what can we conclude from this? If we know more of this sequence, and we know that species 1 has the sort of original version of the DNA, we know that species 1 comes up first on this cladogram. And then a mutation occurs. A mutation in base 4 changes a C to an A. And sometime after that, we see species 2 and species 3. Pretty easy, right? Well, what if we have a similar situation, but now we have 4 species to compare? And one of them has what we know to be our original sequence. Species 2, 3, and 4 all share a mutation on the 4th base that changes a C to an A. But species 3 and 4 have another additional mutation that isn't shared by species 2. Their 6th base has gone from G to T. Well, what would this look like on a cladogram? Again, we know that species 1 is the original. We have other evidence that that's the original sequence. And we know that at some point a mutation in base 4 turned a C to an A. Species 2 arises after that, and species 3 arises after that. But for species 3, there was an additional mutation that occurred, which was that base 6 mutation, changing the G to a T. Whoops. We know that both species 3 and 4 appear after that mutation in base 6. Pretty elementary so far? So let's say we have something with a little bit less information. 4 species were not given the DNA sequence, but we're told that there are some number of differences in gene X. And we're comparing everything to some reference species that we know has what we'll call the older or more original version of the gene. We know that when we look at this gene, species K has 1 difference compared to the reference species, L has 5, species M has 4 differences, and species N has 2. What can we get out of that? Well, we can't answer all our questions with this information, but what we can say for sure is that K and N are probably more closely related to one another because they only have 1 or 2 differences from the reference species. They also probably share a common ancestor more recently than the reference species, so their little branch of the cladogram is going to come off somewhere about there. What about L and M? We know that L and M, because they have 5 and 4 respectively, differences from the reference, have to be more closely related to one another than they are to K and N or the reference. Because they have more differences from the reference, we also know that their common ancestor with the reference species is probably further in the past. DNA isn't the only chemical that we can look at when we're looking at chemical evidence of evolution. We can look at proteins, such as hemoglobin. The protein that moves oxygen in the blood, that's hemoglobin, it's used in every single vertebrate on the planet, and even a few invertebrates. Human and gorilla hemoglobin is only 1 amino acid different. So, this has been a very brief introduction to how to use chemical evidence to build a cladogram. We're going to work with it a little bit together, and then you're going to do some questions. That's it.